Welcome to Conservation of Genetic Resources. Today we are going to be looking at some definitions and then conservation of genetic resources through mapping of existing biological diversity, which is one of the conservation strategies. Conservation of an animal genetic resource is the sum of all actions involved in the management of animal genetic resources, such that these resources can be utilized and developed to meet immediate and short-term requirements such as for food and agriculture, while also maintaining a diversity harbor that can meet longer-term needs for future generations. So if it is conservation of plant genetic resources, the same thing. If it's conservation of forest genetic resources, the same thing. So conservation is simply, what are all my strategies that we are using to manage genetic resources that are available for now and for future. We conserve genetic resources because of the threat of genetic erosion, which is usually the loss of individual genes and gene combinations from a species by the replacement of local varieties by maybe newer ones. Then maybe there is emergence of new pests that are consuming the existing diversities, resistance to pests or to pests of pests to pesticides, so that they consume our genetic diversity and they are not easily destroyed. And then other environmental changes and natural maybe calamities that could cause changes in genetic diversity. And now we usually will have two ways of conserving genetic resources. One of them is what we call in situ, and the other one is what we call ex situ. In situ and ex situ. In situ consists managing populations on site, dynamically involving their natural environment. So this is where we do conservation activities, where that particular genetic resource, is it a forest, is it an animal, is it a plant, in its natural environment. So in situ methodologies will include conservation of natural populations in nature or on farm conservation. Then ex situ usually will consist of conserving individuals or populations out of their natural environment. So ex situ, we have the gene bank methodologies. So conservation in seed banks, what we call in vitro conservation, cryopreservation, and of course through international policies that now will do what we call ensuring fair and equitable sharing of benefits that are derived from these genetic resources for present and future. So usually we are going to have international policies like the Nagoya protocol, which was made in the 2014, and it is usually there to define and protect the owners of genetic resources, such that if you find a genetic resource within a certain locality or geography, this protocol is able to ensure it defines and protects who the owners are, to ensure that they have access and the benefit in sharing. Like they would have maybe a certain animal only found in a certain geographical region in the world, then this protocol can help define the owners as those within the geographical area, and even if that animal is utilized elsewhere, the access and benefit falls mainly on where it was naturally found. So we have those two methods. So I have just given other examples of in situ, for example, where we are having on-site conservation in their natural habitat, or on farm, such as botanical gardens. We can have in national parks, sanctuaries, biosphere reserves, marine reserves, and wetlands. Of course, we've said that these are now the two main ways of preserving. We have in plants natural reserves and wild nurseries. In ex situ we have botanic gardens and seed banks. Then we also have cultivation practice, which is good agricultural practices that also helps us to do conservation strategies. So why do we conserve genetic resources. One of them, we need to keep potentially useful genes and gene combinations. We need to keep them. We are only now beginning to understand the complexity of genes because of the era of molecular biology, biotechnology, various technologies like bioinformatics that helps us to do various models of our genomes, and also the sequencing techniques. So this is the time we are now able to understand the complexity of genes and how they interact, and therefore we risk losing genes of value if we do not have any means in which you can genetically conserve. So some examples is that non-intensive agricultural production systems may not want genes of prolificacy, such as Burola gene in sheep, which increases the number of lambs per mating. Then hyperprolific lines of sheep and swine are of value also in modern production systems. We have porcine stress syndrome in swine can lead to undesirable quality in pork, but the condition has been associated with faster growth. So DNA tests now are able to allow us to select directly against these allele linked to this condition. So using whatever genetic information that is already there, we can be able to do some of these selections against undesirable traits. Of course, DNA tests have now been developed and commercialized to allow us selecting, for example, beef cattle directly for meat tenderness. Remember we talked about how you can actually select breeds just for meat. Using the calpacitan gene, the frequency of this gene varies among breeds. So you can see that we are able to keep potentially useful genes, and even those that are not useful, we are able to eliminate them, like in the case of porcine. Another reason is that we need to take advantage of hybrid vigor, what we call heterosis. Heterosis is the increase above the average of the parent stocks obtained by crossing genetically diverse breeds. So crossbreeding is practiced widely in swine, sheep, and beef production. And if only a few breeds are kept, the opportunity to develop good crosses is lost. So we are saying hybrid vigor is where you're able to cross extremely diverse breeds for you to get certain advantageous breeds. So we need to keep these genetic resources so that we're able to cross them to get even better animals, better plants, better organisms that can be able to survive even in future when we have uncertainties of environment. And to overcome the selection plateaus. So a selection plateaus occurs when genetic variation is lost. I remember we talked about maybe some animal has been wiped out by a natural calamity and diversity has been lost. And even if that population was able to grow again, we are still not able to have more genetic variations. So in such a situation where we are having a plateau in terms of genetic variation, where we have no further change is possible because animals are genetically alike. If genetic variation exists in other breed crosses, this can be overcome. So if we have now been able to store or have forms of variation within other breeds, then it is possible for us to maybe recover our loss of genetic variation. It also helps us to provide insurance policy against, I have kind of mentioned this, we all need insurance in one way or another because of eventualities that are not known. So we are able to provide insurance policy when we conserve genetic resources through situations of climate change, situations of spread of disease, then changing availability of feedstuff. Maybe there is no more enough food. Maybe social changes such as issues of animal welfare and environmental sustainability. Maybe at some point we'll no longer be allowed to have animal models for some reason. But if we have already stored genetic resources somewhere, then it will not affect. Then also selection errors. A widely used sire may spread a genetic disease throughout a population before the problem is identified. So sometimes maybe you have done selection of the breeds you want, but what if you spread a genetic disease and you're not able to trace? Because sometimes you may not be able to trace, especially if you have already crossbred several animals. So such errors of selection can be corrected if we have had a genetic resource kept. We also conserve for cultural purposes. Of course, human history is closely linked to agricultural practices and the use of particular breeds, poultry breeds such as the Bird, Plymouth Rock, the heavy horse breeds such as the Percheron and Clydesdale were common on farms. These breeds are now used on living history parks or living museums because we do not have them today. Even the case of dinosaurs and other animals that we used to have, and now we use them for education and tourism in our museums and parks. We also conserve genetic resources for research, very critical for scientists. We control or unselected lines are used to measure genetic progress in selection. With time, we will be able now to keep monitoring how our genetics changing with time. So identification of specific genes which regulate traits such as product quality and health is made easier by comparing different groups of maybe resources that you have already kept, so that those that were kept a long time ago can be compared to now. What are the genetic progresses that we have had? Then economic evaluation of breeding programs now includes sociological aspects as part of a focus of sustainable rural development. So research into the role of minor breeds in such production system is also required. We have two major goals of conservation. One is to keep genetic variation as gene combinations in a form that can be easily recovered. Live animals may be appropriate for some situations. Other situations we do cryopreservation. We talked about very low temperatures, like of that of liquid nitrogen, which is -196. We do cryopreservation, that's extreme freezing of sperms, ova, or embryos of many different species. And also new tissue culture technologies are also showing promise in us being able to keep these gene combinations. Then we also do it to keep specific genes. As gene sequences linked to specific traits are identified and defined, we'll be able to save those DNA portions of interest. So we're saying the first goal is to keep all genetic variation. That's why you're using a whole embryo, a whole sperm, or even a whole live animal. But sometimes we just need specific genes, not gene combinations. Maybe you just need a gene that is responsible for a certain characteristic. So those two are the goals, either gene combinations or specific genes. We have the steps necessary for conservation. As we plan how to carry out genetic conservation, we go through certain processes that are important, and the very first is inventory. So definition of a breed as endangered depends on factors such as the number of breeding males and females, what are the overall numbers, number of subpopulation, and trends in a population size. It is thus important for us to monitor numbers and changes in numbers on an ongoing basis. An inventory just means you're every day or at constant periods of time you are doing mapping of what are the numbers of certain organisms, certain plants, certain forests that exist, because we have forest diversity, marine, aquaculture. So you kind of do a map of what are the numbers of this population, their subpopulations, their overall numbers on an ongoing basis, to understand how they are maybe changing with time, and therefore you can be able to note if they are increasing, if they are decreasing, if they are becoming endangered, or many other factors that could be unfolding. Then you have what we call evaluation. So stocks must be characterized for phenotype and genotype using new technologies as appropriate. So now here we want to define phenotypes and also define genotypes. So gene mapping approaches such as testing for single nucleotide polymorphisms help track things like ancestry and determine genetic distance of one group from another. Then of course phenotypic, or the appearance performance evaluation, must be standardized and carried out in the environment in which the stocks might be used. So when you have had genetic resources, you need to keep evaluating what are their phenotypes, what are their genotypes, then link them to the we call it family trees. You can be able to do or to map ancestries in gene mapping. Then we have choice of breeds for conservation must include cultural reasons, potential value and threat of extinction. So of course whatever you choose to conserve cannot be literally everything, because there are some organisms or plants that may not need much conservation. So you must also check in terms of what are the cultural reasons behind certain organisms. Maybe they have been highly rated for maybe cultural practices, and then maybe potential value, something that is useful in maybe increasing breed or has extraordinary characteristics, or maybe there is a threat of extinction from that particular organism. These days we have new mathematical techniques and economic theories that can help us assess the risk of loss and also to check for potential benefits of certain organisms. Saving pure breeds preserves that breed characteristic and make it readily identifiable animal. Crossing several breeds to produce composites has the advantages of also saving genetic material from all while reducing upkeep costs. However, the cost of total genotype of each breed may be lost. So we are able to choose pure breeds which will preserve a certain breed's all their characteristics, but we can also do breeds that are not pure, or those that have been crossed, okay, so that we have a composite advantage, that is a combination of many different genetic characteristics that has an advantage. Although in this case we are going to lose the genotype of each breed because we have done a combination. Some of the total genotype of each of what you crossed is likely to be lost. Then we have what we call preservation. Populations can be saved as live animals. I remember we talked about that. That one is one of the goals of how we preserve. We can use live animals as a way of preserving. Get one animal from the environment, or two, a female and a male, and put them somewhere in a quarantine. Of course, this is expensive, and unless the breed can be used for production, it is not likely to succeed. That means if it cannot reproduce, then this can be a difficult process. Development of niche marketing schemes emphasizing the traits of a particular breed can be successful. Linking breeds maintenance with tourism and education. Of course, once we preserve a whole animal, we are also doing what we are calling linking it to tourism and education for students and researchers. And then DNA collection, of course, we said this is another way. You can just extract the DNA. So the potential exists to use DNA and cloning to redevelop breeds. But the technology is still new and costs very high. Of course, the technologies of cloning are very high, not highly used, but we can be able to extract DNA either way and have it stored. Whether kept as live animals or as frozen material, more than one location is needed. Yeah, because of natural disasters and accidents and changes in financial resources as a result of instant loss of the stock. So to avoid stock loss, you cannot put a DNA of an animal or a plant in just one location. You have to redistribute it in different places in case there will be a natural. If you put it in one lab, what if the electricity gets, of course there is backup, but what if there comes an issue of electricity, your power banks are not working, then it would result to loss of that DNA that you have collected. Of course, we have cryopreservation. Once you have collected DNA and stored it appropriately, then you need also to do what we call cryopreservation of other. Here we do cryopreservation of semen, ova, and even embryos. It has been done successfully for cattle, but of course it is difficult for some species, but we keep researching and exploring. So for those species where cryopreservation is routinely practiced, a national center for monitoring and maintaining frozen genetic resources is needed. So who is involved in genetic resource management? We have commercial animal or plant or forest. So this stands for everyone. So the commercial industries, and of course we have individuals. So the commercial industry must emphasize traits that have economic value. Of course, we have to also check what can actually bring in even cash flow and in the short-term future. So those that will have economic value in the present and also in the future. Industrial breeders keep genetic stocks as necessary to satisfy that need. Like we have the Kenya Breeders Association, mainly for farm animals. So that one is a commercial animal industry. Then individuals, we have private producers who keep these stocks of minor breeds either as a hobby or as part of a farm enterprise, and the emphasis may be on phenotype, and small populations may lead to reduced genetic diversity, and the stocks that they keep are subject to loss as a producer's situation changes. So this one is not like permanent, but there are individuals who have taken part. I have even gone to national parks that are for just specific people, not really owned by government. Someone just has it as a hobby or as a farm enterprise. We also have conservation groups, breed organizations, and private producers obtain funding. Yes, now these ones, they have gone ahead, they have obtained funding, and they provide resources and information on conservation of animal genetic resources. And then you will have it having members, maybe the farmers of, if it is a conservation group for cows, you're going to have dairy farmers being part of that conservation group, and then they are going to involve other scientists. Then you're going to have breed associations and commodity organizations. Sometimes they help them improve their breeds so that they can also sell. What maybe we can understand easily is the breeds, the conservation groups that are used for plants, like maybe maize farmers. You find that maize farmers are in a specific group, so usually they are going to have improved maize with time, so maybe this year they have improved on this. Maybe the maize are heat resistant, pest resistant, etc. So they keep improving because they have scientists among them, and they also help in commodity sale. They sell for them their plants, their crops. They provide a market for the sale of those products. That's a conservation group. Of course, we have government. Limited funding is available from national governments for conservation. Mainly we get funding from private organizations. Then agriculture research stations and many universities also keep stocks of livestock breeds. Although these days maybe they have been minimalized. So you find that government, in terms of universities, in terms of research stations, like now maybe with medical, we can have places like Cambridge, maybe University of Nairobi, because they have a medical wing. And then I've also seen, I don't know if you research, maybe we are going to see as people present on national parks and reserves. We have different institutes of universities, like there is a University of Nairobi Marine Studies institution in Mombasa, where we will have now students who are pursuing marine studies, being in a place where there is several stocks of existing marine diversities, so that they're able to study as well as continue conserving and study of also genetics and phenotypic characteristics. We have what we call FAO, for agriculture, food and agriculture. United Nations plays a major role in assisting individual countries with conservation programs and provides a forum for international consultation and planning. And of course, even their website will provide a lot of information and methods and resources, how to conserve, what are the best practices that can be undertaken by nations, what other people elsewhere in other places are doing, because we keep learning from each other. Then of course we have conferences that are organized, such as the World Congress on Genetics applied to livestock production, providing a forum for scientists involved in genetic diversity. They also need learning as scientists pursue many various methods of conservation. They have a forum where they can learn what is going on and ways in which they can keep doing what they do, and whenever possible funding may be made available to allow them who are from developing countries to attend, like most of the African countries. Yes, that is conservation of genetic resources, simply looking at the definitions, what are the goals, why do we do conservation, and who is involved in conservation strategies.